Penicillin, the beta-lactam antibiotics. If we talk about the history of the penicillin, it was clearly seen that once a scientist working at the St. Mary Hospital in London discovered that a culture plate on which the staphylococci were being grown had become contaminated with a mold of the genus Penicillium. He made this observation while working, and he made the crucial observation that bacterial growth in vicinity of the mold is in pure culture is inhibited, and while on other part there was a huge growth of the bacteria. He subsequently isolated the mold in the pure culture and demonstrated that it produces an antibacterial activity which he called penicillin, and that work of scientists help a lot to the humanity, and that great scientist of the world was known as Alexander Fleming. Now, if we come to the structure of the penicillin, it consists of the 6 amino penicillinic acid structure nucleus, which consists of the one thiazolidine ring structure, and the other part is beta lactam ring. To the beta lactam ring, a secondary amine group is attached, which consists of R substitute, which determine whether the penicillin is amoxicillin, in which R substitute will be containing this structure, containing a benzene ring-like structure with the CH attached with the amine and OH. And if OH in this structure is eliminated, the following structure will be ampicillin. And if we make a structure of benzene ring attached with the OCH2, then this penicillin containing R substitute like this will be penicillin V, a natural penicillin, and if O in this is eliminated, the following structure will be penicillin G. This is also a natural penicillin. The aminopenicillinic acid nucleus or the penicillin pharmacological antibacterial activity depend upon the structural integrity of this structure, and beta lactamase by hydrolysis call its hydrolysis and produce 6 amino penicilloic acid structure. 6 amino penicilloic acid structure will not contain the pharmacological or the same antibacterial properties, and uh, it will not maintain its integrity. And now if we come to the mechanism of action of the penicillin, which is mostly a ghost of for the medical students, and first of all we will discuss the structure of the bacteria. Bacteria by transpeptidation uh, produces the cross linking. If we come to the bacterial structure, whole structure, it will have bacterial cell wall, periplasmic space, cytoplasmic membrane, and at the end cytoplasm. In the periplasmic spaces, penicillin binding proteins are present, which are enzymes for the attachment of the peptides or the penicillins. Bacterial cell wall, if we discuss the bacterial cell wall structure, it consists of the it will maintain the structural integrity of the cell and consist of the peptide ring and polysaccharides, and these two structures will make as a whole the peptidoglycan. And now if we move to the structural integrity, that how these cells are maintained, this consists of the uh, N-acetyl glucosamine, N-acetyl muramic acid structure. Polysaccharide consists of, and to the N-acetyl muramic acid structure, 5 peptide ring is attached that terminate um into D-alanine, D-alanine, and during the cross-linking process, the D-alanine is eliminated by the penicillin binding proteins by its attachment, and by the elimination of the D-alanine, the cross-linking occurs transpeptidation by the process of transpeptidation, and the process will be like this. And cross-linking, due to cross-linking, the structure will be like this, and this will be helpful in the formation of the cell wall structure. And now if the antibiotics like penicillin is attached, it will attach to the transpeptidase, and no cross linking occur and no bond formation, due to which the structural integrity of the cell is damaged. No cell wall cross linking occur and the cell wall synthesis inhibits, and the cell wall integrity decreases, and cell will not protect from the outside plasmic osmotic pressure and the outside pressure, and as a result cell wall inhibition, and the cell will die. Also, in combination with the cell wall inhibition, the cell death mechanism is also occurred by the lysosomes, which help this process and synergize this process by their action. And the R substitute will determine the pharmacological, biological, antibacterial properties of the penicillin and helpful in the classification of the penicillins. And basically, on the pharmacological and the biological antibacterial spectra, the penicillins are divided into 4 groups. First is the natural penicillins, consisting of the second is the semisynthetic penicillins. Third are the anti-staphylococcal penicillins. And the fourth are extended spectrum or anti-pseudomonal penicillins. And now, if we come to the natural penicillins, these are penicillin V and the penicillin G. Penicillin V consists of a chain ring, as we already discussed, in which a benzene ring OCH2 penicillin V, and if O is eliminated in this R structure, then it will be penicillin G. If we discuss the spectra of the natural penicillins, they act on the gram positive, gram negative, and the anaerobic, and their hydrolysis occur by the beta lactomases. Penicillin G are the drug of the choice for the streptococcus, staphylococcal, but not producing the beta lactamase. And they are drug of choice for the pneumococci that are also penicillin susceptible, and also syphilis. Now, if we come to the semisynthetic penicillins, these may be amoxicillin and the ampicillin, the most widely used antibacterial of the penicillin class. And they are both collectively known as penicillins. Uh, their spectra is gram against gram positive, gram negative, cocci and the rods. In comparison to natural penicillins, they are not against gram negative cocci. The rods, these natural penicillins are not gram negative rods. These are also gram negative rods active. If we come to the widely used combinations, brand names, amoxil, the amoxicillin, and the augmentin, the widely used combination, it is amoxicillin and the clavulanic acid and the unacin. Unacin is amoxicillin, ampicillin plus sulbactam. If we come to the antistaphylococcal penicillins, these are the methicillin and the nefcillin, and their spectra are the against uh, beta lactam producing or the not producing staphylococcal and the streptococcal bacteria, and their drug of choice was the endocarditis, but the resistance of the methicillin is a rising problem, so we have to confirm before using this the either the infection is methicillin resistant or not. Now if we come to the anti-pseudomonal penicillins, or the also known as extended spectrum penicillins, these include mainly two classes or of carboxypenicillins and the uridopenicillins. Carboxypenicillins usually contain mostly used two drugs, carbanicillins and ticarcillin. Ticagrelin is mostly used with the combination of the tolbactam and the beta-lactamase producing drugs which enhance their activity. Amoxicillin is also used with the combinations like this, and the other is penicillins. Uridopenicillin, like piperacillin, this is also used in the combination with the beta-lactamase producing which enhance its activity. Uh, and this is the most widely used combination. And now if we come to the adverse effect of the penicillin, there may be hypersensitivity, allergic reaction, angioedema, anaphylactic reaction, and the edema joints. And hypersensitivity reaction include the anaphylactic reactions. And if the patient, the most important considerations are if the patient has renal failure, penicillins are contraindicated because they may cause seizures, and this is most dangerous than not using the drug or bearing the common cough and the other problems. So consider this. And if now we come to the other penicillin, and if we talk about the ADRs of the other penicillins such as ampicillin and amoxicillin, skin rashes are the most common, especially in the setting of the such as Epstein-Barr viral disease. And when the piperacillin plus tolbactam combination, usually known as zosine, is given with the vancomycin, acute kidney infection chances are increased as compared to other beta-lactam combinations used with the vancomycin. And if nefcillin is used, the chances of the neutropenia and the interstitial nephritis increases. And when the oxacillin is given, hepatitis chances are increased. And also when the uh, penicillins are given in the high doses, the chances of the GIT disturbance or the GIT problems increased. And if the methicillin is given, the chances of the interstitial nephritis increased. That is the reason why it is not used most commonly. And now if we come to the resistance mechanisms, resistance is basically due to the four mechanisms. First of all, decreased permeability of the penicillin in this cell through the porins. It may be due to the decreased number of the porins or altered structure, and secondly, it may be due to the increased efflux mechanism due to the production of the more efflux pumps. And the third mechanism is the altered structure of the penicillin binding proteins, due to which uh, the drug may not be attached to it, and the transpeptidation reaction may not be stopped, and the cell wall synthesis will not be stopped, and the cell um, continue its growth. And the fourth mechanism is increased degradation of the penicillin by the beta lactamases. And so these are the four mechanisms of resistance. Now, if we talk about the preparations of the penicillins most widely used in the market for the medicinal clinical purposes, these are the amoxil, which is generically amoxicillin, and augmentin, which is amoxicillin plus clavulanic acid combination, and the third preparation is Ionacin, which is generically ampicillin plus sulbactam combination, and the fourth preparation is Zosine, which is a combination of the uh, piperacillin plus sulbactam. And now, if we talk about the general uses, these are mostly used for the pneumococcal, meningococcal, staphylococcus, and the endocarditis, also for the cough, as augmentin. And augmentin is also used for the skin problems. And after all these, mechanism of action, resistance, pharmacokinetics, ADRs, uses, I have a medical case for all of you. Hope you love it. And this case will enhance your medicinal ability and the medicinal practice. That is, if a patient has a pneumococcal plus meningococcal infection confirmed, we prefer penicillins. But if the patient has penicillin allergy, to which class you will move to treat that patient? Please give your feedback in the comment section. And if you like our video, don't forget to subscribe and press the bell icon and like to encourage us to make more videos. Thank you.